previously on DNA and RNA,
but will also reveal the identity of the true killer of our poor departed Mr. Bones.
But there are 4 different nitrogenous bases: guanine, adenine, thymine, and cytosine. You see, cytosine can form 3 hydrogen bonds with guanine, and adenine can form 2 hydrogen bonds with thymine. If we represent the strands as arrows, with the arrowhead at the 3' end of the strand, we can see that the strands in a DNA molecule are organized anti-parallel relative to each other.
Objection. How do you even know what he's saying is true? Professor, you say that the deoxyribonucleic acid, or DNA, is a recipe for life. But how do we know you aren't just throwing around your fancy science word in a big smoke screen to clear the accused, Colonel Custard, of this crime? Can you offer proof that DNA is the molecule responsible for transmitting heritable traits in organisms? Answer me that.
Oh yes, I certainly can. There's lots of evidence, but let me just summarize the work of Frederick Griffith and Oswald Avery. Their labs provided strong evidence that DNA is the molecule of heredity. Griffith was studying the bacteria that causes pneumonia. He observed 2 variants of the same bacteria under the microscope. One had a smooth outer appearance. Let's abbreviate that bacteria as S bacteria. The other had a rough appearance. Let's abbreviate those bacteria as R bacteria. If he injected a mouse with S bacteria, the mouse died. If he injected a mouse with the R bacteria, the mouse lived. We would later discover that the S bacteria is smooth because it has a protective coating which helped it survive in the mouse, whereas the R bacteria lacked a coating and was susceptible to the mouse's immune system. If Griffith heated the S or R bacteria, it killed the bacteria. Not surprisingly, a mouse injected with the dead bacterial parts did not die. Oddly though, if he mixed dead cell parts from S bacteria with living R bacteria and injected a mouse, the mouse died. Isn't that peculiar? But that's not all. When he examined the dead mouse more closely, he found S bacteria, not R, inside the corpse. Somehow, the R bacteria had transformed into S bacteria. This transformation was permanent, meaning it was a trait that was inherited from generation to generation. Using the mysterious new S bacteria from the dead mouse in a new injection experiment also produced a dead mouse. That meant that the R bacteria had permanently been changed into S bacteria. Oswald Avery dubbed Griffith's mysterious substance that transformed the R bacteria into S bacteria as the transforming principle. He decided he and his lab would determine the identity of this substance. I should probably provide a little context for this time in scientific history. At the time of Avery's experiments, most scientists believed DNA to be uninteresting compared to proteins and less complex than even carbohydrates or lipids. For instance, there were 20 known amino acids, but only 4 types of DNA nucleotides. Surely, a molecule of such simple complexity couldn't be the molecule of heredity. Avery spent many years purifying the transforming principle, then tried to characterize which type of molecule was responsible for transforming R bacteria into S bacteria. To identify the molecule that could transform the R bacteria into S bacteria, Avery and his team devised a series of clever experiments. By spinning the purified sample very fast in a machine known as a centrifuge, fats were eliminated from the sample. Sample treated in this manner could still transform R cells into S cells. Shockingly, treating the sample with something that degrades protein did not affect the ability of the purified sample to turn R cells into S cells. Treating the sample with a substance that degrades RNA also did not affect the transforming principle. However, treating the purified sample with something that degraded DNA eliminated the R to S transformation. Avery concluded that the transforming principle was DNA. Today we know that permanently changing the characteristics of an organism can be accomplished by changing its DNA content.
Well, what about your assertion that you know the structure of DNA? You yourself said it's a microscopic molecule found inside a cell. If that's true, how can you possibly know what it looks like?
Oh, actually, that's another fascinating story. James Watson and Francis Crick devised a model of the structure of DNA based on the evidence produced by several different laboratories at the time. Examining X-ray images of DNA revealed that the molecule had a helical or spiral shape. Data from another lab indicated that there is a 1-to-1 ratio between adenine and thymine. The lab also demonstrated that there is a 1-to-1 ratio between guanine and cytosine. By using cardboard cutouts of the bases, Watson realized that 2 hydrogen bonds could form between A and T, and 3 hydrogen bonds could form between G and C. Interestingly, Watson originally predicted that there were only 2 hydrogen bonds between G and C, but we know now that there are actually 3. Even a Nobel Prize-winning scientist is wrong sometimes. In order to reconcile the X-ray data and Watson's model of the ratio between bases, Crick realized that the DNA strands had to be oriented anti-parallel to one another. This led the two of them to postulate the famous double helix structure of DNA. A helix is a cylindrical spiral. A double helix is basically just 2 cylindrical spirals. Picture DNA as a ladder with backbones made of phosphate groups and sugars, and rungs of nitrogenous bases held together by hydrogen bonds. Then twist the ladder around an imaginary central axis. The structure of the molecule looks a little like a spiral staircase, not unlike the one on which the body of the victim was discovered. James Watson, Francis Crick, and another scientist named Maurice Wilkins were awarded the 1962 Nobel Prize in Physiology or Medicine for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material.
Madam Prosecutor, if you're done attempting to rattle my witness, I'd like to continue. Now, Professor, you provided us with quite a nice foundation about the structure and function of nucleic acids. In fact, I wouldn't be surprised if we'd all be able to pass a biology test after this. But let's return to the case at hand. How can DNA be used to vindicate the innocent and catch a criminal?
Well, you can think of the different DNA bases as letters in an alphabet. The DNA alphabet is just a simple alphabet with only 4 letters. Letters can be combined to form different words, sentences, and paragraphs. Likewise, the order of DNA base pairs determines what biological molecules can be produced, and in turn, the characteristics that makes each of us unique individuals. There are approximately 3 billion base pairs in each human cell. With the exception of twins, the chance that a sequence of DNA for any one person is exactly the same as another person is pretty much impossible, simply due to the variation present from person to person. Consider that there's a 1 in 4 chance that someone has the same base pair at a given point in a piece of DNA as another person. There's another 1 in 4 chance at the next base pair, and so on. If you examine enough pieces of DNA, it becomes statistically impossible to mistake one person's DNA for another person's. My lab compared DNA sequence from Colonel Custard, the blood found at the scene of the crime, and the victim. We found no DNA from Colonel Custard at the scene of the crime. However, we did find DNA from the blood of another person attending the conference mixed with the victim's blood.
Thank you for your expert testimony, Professor. Ladies and gentlemen of the jury, you can see that the work of Frederick Griffith and Oswald Avery establishes DNA as the molecule responsible for transmitting heritable traits. James Watson and Francis Crick developed the double helix model for the structure of DNA. In short, DNA is organized as a twisted ladder with phosphate groups and sugars